All right, so I'm going to be talking about patterning today and developmental patterning, but we're going to start by blowing bubbles in sand. And this is not meant as a literal example of anything I'm going to be talking about today, but more as a warm-up for the kind of patterning I'm talking about today. So what you're seeing is a video from YouTube where we've got these two reservoirs, we're blowing air into them, and you can see that the output of the system is very different between these two different reservoirs, where really the only difference is how much we filled them up. And I like this example because really what you're looking at is an emergent product of this system. Okay? These are not specified differences, they're emergent. And that's the kind of thing that appeals to me about auxin transport. And I'm going to return to this water analogy throughout my talk today. So if your brain sort of wanders off and you wonder what I'm talking about, just remember we're talking about flows. We're talking about emergent properties of things like water. Okay? But of course, to the developmental biologists in the audience, it's no surprise that a lot of development is not very specified. It's actually an emergent output of interactions between lots of different components. Okay, so I'm going to start by talking about why emergent patterning might be particularly important for plants. We're going to do a little bit of evolutionary history of plants, and then I'm going to show you how my lab is using comparative genetics, comparative genomics, phylogenetics, computer modeling, and a lot of imaging to try to understand this core mechanism of development in plants that I really think leverages emergent properties. Okay, so I'm going to be talking about the seed plants today. All the data is from the seed plants, but we're going to walk through a little bit of evolutionary history. And what you're looking at here is a phylogeny of the eukaryotes. And as we walk up here to the seed plants, we're going to go through several layers of their evolution and really examine the constraints on what it means to be a plant and why I think emergent properties are important. Okay, so there's at least 6 independent evolutions of complex multicellularity in the eukaryotes. All of these guys started with a single cell ancestor, and there was lots of independent evolutions of multicellularity, including up here in the lineage that gave to seed plants. So just keep in mind, this is an independent innovation of three-dimensional growth. It's an independent innovation of tissue formation, and so it's a bit different. But I think you'll see some overlaps. Okay, so along this trajectory, first we started out as a single cell, then through the endosymbiosis of a cyanobacteria, there's a commitment towards photosynthesis. Okay, so we've got a single cell, it's now autotrophic, and that commitment towards autotrophy really makes environmental sensitivity an important part of what it means to be a plant. We can't simply stand up and walk away. We must move towards the light and things like that. So autotrophy really defined plants. Also coincident with this is the evolution of a cell wall. This is a polymer of sugars that surrounds these cells, and this acts as a physical constraint to cells and as a physical constraint to development. And then along this lineage here in the streptophytes, which include the charophyte algae and the embryophytes, you get the evolution of plasmodesmata. And plasmodesmata are really fascinating structures, but in short, after cell division, these two daughter cells maintain a cytoplasmic and even ER connection between the two cells. So there's a piece of contiguous membrane between the two daughter cells. Once again, a very strong physical constraint. And of course, I'm leading up to this typical thing that a lot of people say about plant development is cells do not move by and large. And the reason for that is the shared symplast. So symplast is this term that describes the shared cytoplasm of a whole bunch of cells. They're all, by and large, connected to one another. And then here, up into the land plants, you get the evolution of three-dimensional growth. The ability to make cell division planes in 3 dimensions. And here we are. I love these sort of reconstructions. We're 400 million years ago, some sort of marshy plain, and this is a fossil called Cooksonia, which is an early vascular plant. And what I want to point out to is a couple of the characteristics that are maintained in seed plants today. Those include the commitment to autotrophy. These are photosynthetic organisms. There's photosynthetic tissues. You can see a branching axis. But most importantly, for what I'm going to be talking about today, most of the growth occurs from the tip. Okay, so plants grow apically, and there's a population of cells that are sending differentiated tissues behind them as they grow. And I'm going to point to these fossils. So these are actual fossil tracheid elements from Cooksonia. And these are xylem vessels, okay? So dumb, dead cells, a tube of dead tissue that is involved in moving water apically in the plant. And this, to me, is an important problem because as that plant grows apically, it must maintain these developmental connections to an existing system of transport that's dead. Okay, so it's really important to maintain that connection and is a further sort of constraint on how we pattern that tissue. And I'll just point out that this basic habit has been wildly successful. Okay, so the vascular plants, which include the lycophytes, ferns, gymnosperms, and of course the angiosperms, have just a tremendous species diversity and tremendous ecological success. So move forward 400 million years, we have the modern meristem. This is my favorite meristem, the Brachypodium spikelet meristem. It has a lot of the same characteristics of Cooksonia. This idea of apical growth is very important. So most everything is growing from a population of cells right here, and differentiated tissues are down here in the bottom of the plant. This SEM is lovely because you can really see how these cells are packaged in like bricks, constrained by that cell wall. Physical constraint to cell movement, and then just keep in mind that they share a symplast. A lot of these cells are connected by plasmodesmata. So up here is the stem cell population, which I'm sure you've heard a bit about in the department, and that stem cell population can branch, creating new branches, and each of these meristem tissues can make organs. And this is the organ I'm going to be talking a lot about today, the lemma, which is a leaf-like organ in the Brachypodium spikelet. And this is where we're looking at a lot of the developmental patterning and morphogenesis that we're trying to study. Okay, so once again, keep in mind that at the base, there's all these mature differentiated tissues that need to be plumbed in with all the new developing tissues. And despite that, up here, we're integrating all kinds of environmental inputs. So there's a degree of plasticity in the meristem that needs to be maintained to respond to the environment, and yet all the patterning must connect to differentiated tissues below. So if I can just show that in my cartoon here, we've got the addition of cells apically and differentiation coming behind. Okay. So one of the things is, how do you pattern? How do you pattern this type of tissue where you have all these physical constraints, where you have the need to connect? And I'll tell you quite simply, it's with the flow of a signaling molecule. The movement of auxin from the top of the plant down the plant, the active transport through tissues, the diffusion through tissues creates pattern, it creates polarity, and it connects differentiated tissues with developing tissues and creates this vast system of long-range communication in the plant. And this, I think, leverages emergent properties to give it its plasticity, and that's what I love about auxin transport. Okay, so this is where I return to my water analogy, and I'll just point out that one of the things that created these patterns was a positive feedback, the positive feedback of erosion. The more water you move through a landscape, the more you erode, the more you erode, the more water you're going to move, and you end up with the Grand Canyon. But because of the positive feedback and the emergent nature of erosion, really the Grand Canyon wasn't specified as it is. It's got an order, it's got major axes and tributaries, accessories, but you would be hard-pressed to say, oh, it was specified from the base or it was specified from the tip. No, it was an emergent pattern that came out of the flow of water through the system. And this, I think, has been capitalized to make plants as diverse as they are, and I think during developmental time has been capitalized to give plants a degree of phenotypic plasticity that they need with their autotrophic habit. Okay, so how does this work molecularly. Auxin, this signaling molecule, is able to easily enter the cell, but it can't get out. And that's because of a concept called acid trapping. So in the low pH outside the cell, auxin is protonated, but it's a weak acid. So once it ends up inside the high pH of the cell, it becomes deprotonated and charged. And in that charged state, it can't easily exit the cell. Now, I should say that once it's inside the cell, there's a whole suite of nuclear signaling pathways, developmental programs that are set into motion, which I'm not going to be talking about today. Auxin is a tremendously important molecule, and it's pretty much become a joke in the community that it's upstream and downstream of literally everything. It's just comically important. But I'm not going to be talking about that today. I'm going to be talking about the movement of this molecule through tissues and how that mediates pattern. Okay, so it can easily get in, it can't get out. That means that efflux is a rate-limiting step in the movement through the plant. And I'll introduce you to the protein that I'm going to be talking about today. These are the PIN proteins. PIN proteins are membrane-spanning efflux carriers, and they tend to have a polar distribution in a lot of different cell types. And what that polar distribution allows you to do is apply a directionality to auxin movement. So if my auxin is in this cell and it's exported by my PIN here, it's going to be easily uptake into the adjacent cell, and in that cell it might have polar PIN that's directing it yet in another direction. Okay? So that's what PIN proteins do. They link cell polarity and tissue polarity, and they control the distribution of auxin throughout the plant. So I'm going to review a few different important phenomena for how PINs mediate development. And throughout my talk, you'll be seeing polar PIN displayed with these little smiley faces and the transport of auxin with arrows. So one of the important things that PINs do is they can actually move auxin against the concentration gradient, allowing convergent polarization of PIN. So you can see these are apically polarized, these are basally polarized, to actually concentrate auxin into local maxima. Okay? So these local maxima are important in various tissues for positional information. So each of the veins or serrations in a leaf was started with an auxin maxima. Each of the lateral roots in the root were started in an auxin maxima. And here in the shoot, the position of new organs along that meristem started as an auxin maxima. Okay? So that's an important thing that PIN proteins do. The other things that PINs do, called canalization, really is analogous to this erosion concept that I've been talking about before. And that's that they facilitate bulk transport throughout the plant. And they're able to take loosely polarized tissue and canalize that tissue into strong axes of transport. And these axes of transport become the veins, and those veins create a network of connected transport pathways that are throughout the plant, and those facilitate long-range communication. So by long-range communication, I mean that if this bud over here perceives some sort of light or temperature or anything, it can actually affect this bud over here depending on how much auxin is being transported into that shared pathway. So these are great phenomenologies. This is my way of sort of pointing to the fact that there's this large developmental distributed system. It's an environmental integrator, and it's governed by positive feedbacks, but by and large, we do not understand how PIN proteins are polarized with respect to auxin. And this is the question that keeps me up at night, haunts me, quite frankly. How are PINs polarized in response to auxin? Okay. And my inroads into this came from the shoot. Okay, so here's Arabidopsis, this very common plant model. Here is the inflorescence meristem. There's the stem cells. This mutant is actually the namesake of PIN proteins because of this very dramatic phenotype. This is Arabidopsis PIN1. It's a pretty much complete inability to make lateral organs. This meristem is barren. It's not making any of these lovely flowers or primordia as before. But probably one of the key experiments for trying to understand what PINs do came from this experiment, and that's where if you take a little dollop of auxin in a lanolin paste and apply it to that meristem, you can actually recapitulate organ initiation. And this led to the idea that what pin 1 was doing was actually concentrating auxin in the meristem, and that concentration of auxin is necessary for organ initiation. So I'm going to show a lot of the phenomenology of pin polarization in the shoot with this Arabidopsis pin 1 expressing tomato meristem. And these are two different organs at two different developmental stages. This one's younger, this one's older. And what you can see is a lot of polar pin heading towards this spot, this convergence point, where all the polarization is presumably concentrating auxin, and that is necessary for organ initiation. I'm going to mark convergence points throughout the remainder of my talk with these asterisks. And so the idea of a convergence point is associated with concentrating auxin, but then here is one of the central paradoxes of how this works. Here in this slightly older organ, you see a subset of these cells that invert the polarity of their pins. Now all of a sudden, all this pin is oriented away from the maxima, and that's a very confusing phenomenon. This axis right here, where you can see all this basal localized pin, is going to form the very first vein of the plant and plumb this new organ in with all the existing vasculature. And this is canalization, again, the erosion hypothesis. Okay, so this inversion of polarity is one of the things that we've found genetic tractability to study. And I'm finally going to get there soon. Okay. So, but first I'm going to take a brief hiatus to discuss why grasses at all. So I work on a system called Brachypodium, but I just want to point out the obvious, which is that essentially grasses are probably the most important plant group to human survival. We are essentially derived from corn. They make up most of the crops, a lot of arable land. They're important, very important study systems. But I use a model system called Brachypodium. This is Brachypodium here, and the reasons for that are all those things that you like in a model system. It's fast, it's small, it's easy, etc. Great selfing genetics, small genome, but probably most important for what I do, the transformation is just far more tractable in Brachypodium than a lot of other grass systems. And during my PhD, I was able to take this organ called the spikelet and develop this as an imaging system. And I can't think of a better way to do imaging than this spikelet. It's just a wonderful place for live imaging. So that's why Brachypodium, that's why grasses. The other reason came from a lot of phylogenetics, which unfortunately I have to summarize in this cheesy cartoon. But it turns out that when you look at the pin, just a small subset of the pin family in the vast majority of angiosperms, Arabidopsis is simply not representative. And that's because placed phylogenetically sister to the pin 1 clade, so here's Arabidopsis pin 1 up here, is this clade which I named sister of pin 1, and this gene has been lost in Arabidopsis, and in fact the entire Brassicaceae family, which Arabidopsis belongs to. I don't have time to show you the specifics of this gene loss, but suffice to say I really believe it's a loss of a whole gene, and that means that Arabidopsis is a bit weird. There's this sister clade that no one had really studied before. In Brachypodium, that clade is represented, but also grasses have this grass-specific duplication of pin 1A and pin 1B. So during my PhD, it was a simple experiment. We tagged these with fluorescent proteins under their native promoters and see what that sister clade was doing. And what I'll show you in the first part of my talk is that this contradictory behavior where pin proteins are oriented towards the maxima at certain points, then oriented away from the maxima at other points, is actually split between the two different clades in Brachypodium. And that split of these two phenomena gives us power to study how pins are polarized. Okay, so here's the Brachypodium spikelet meristem, and I'll just show you why I love it so much. First, it's very small. We can get lots of these off the plant. The resolution is wonderful. I'm actually staining the cell wall here. We can get the laser very deep into this tissue. the phyllotaxy is disticus. So what that means is that each new organ is placed 180 degrees from the other. And this is probably the most important fact, because then what we get is a nice developmental series of organ initiation. Here's the incipient organ. Here is the organ beginning morphogenesis, and you can tell that because of these periclinal cell divisions. And then here we are, a nice bulge as we go down. So every time I throw one of these meristems on the scope, I get 3 developmental stages, nice developmental series. And it's making the same organ over and over and over again. And this is the first organ of that flower called the lemma. So we're going to look at the lemma during these developmental stages and see what those 2 clades, the sister and pin 1 clades, are doing in Brachydipodium. And from 30,000 feet, when we look at these guys on the scope, I should point out right now that throughout the majority of the rest of my talk, pins will use this lookup table, and when I use this reporter DR5, it's going to be in red. Red is DR5 is an auxin signaling reporter. It's part of the nuclear signaling pathway, so it's a proxy for auxin concentration, a flawed one, but a good one in a lot of contexts. And from 30,000 feet, what you can see is that sister is really abundant in the epidermal tissues, whereas pin 1A and pin 1B really seem to be in the vasculature and internally. So these are all the mid veins of all these lemmas. So one lemma here, next lemma, next lemma, next lemma, moving down the plant. So sister is on the surface, pin 1A and pin 1B are not. Let's look at the polarization dynamics of sister, because it really starts the polarization of this tissue happening. Here in I2, you start to get convergent polarization around where the incipient organ is going to form. Here at I1, you can see nice convergent polarization right around that periclinal division, which is the hallmark of morphogenesis. And, you know, to address the polarization issue, it's a lot easier to see which direction these pins are moving in this tissue than in Arabidopsis, I'll put it that way. But then after sister creates these convergent polarization patterns, it simply maintains that polarization. Now the auxin maxima will form here, the highest auxin concentration is over here, and then it's maintained at that mid-vein tip. Really, in general, sister is oriented towards the auxin maxima almost all the time. Pin 1B, on the other hand, is really the thing that connects the new organ to the existing vasculature. So here's a midvein coming down. Here's that midvein. Pin 1B sort of polarizes the adjacent tissue where sister is doing its little dance over here, and then eventually connects that incipient organ to the rest of the vasculature. The polarization here tends to be away from the auxin maxima. And pin 1A is kind of the boring one, which I'll neglect for the rest of my talk, because it doesn't really show up at these early stages, and when it does finally show up, it's along the incipient vasculature, oriented away from the maxima over here. So you can see these nice little smiley faces, and it really seems to pattern the incipient vasculature. Okay, so I'm going to summarize this localization data here with a cartoon and give you sort of a helpful mnemonic for remembering these proteins for the rest of my talk. Sister is on the surface. It creates those convergent polarization patterns. Pin 1B is broad, and before it polarizes this adjacent tissue and eventually connects this axis of the maxima to the rest of the vasculature. And pin 1A is after and acute. So it creates these very narrow paths much later. So pretty much the rest of my talk will focus on pin 1A or pin 1B and sister of pin 1 because of their early expression. So I've been alluding to this fact for a while now, but the thing that we noticed when looking at this localization is that sister tended to be oriented towards the auxin maxima very consistently, whereas pin 1A and pin 1B tended to be polarized away from the auxin maxima very consistently. And that jived quite well with a couple of existing ideas in the Arabidopsis literature for how pins might be polarized with respect to auxin. And these are called up the gradient and with the flux. And I'm going to show you a computer model where essentially we've taken these 2 different polarization modes and applied them to the 2 different clades and we're able to recapitulate a lot of the polarization dynamics in Brachypodium. And this is a wonderful collaboration with Adam and Shemek from the University of Calgary, who do a lot of computer modeling. And you can follow along with this video that essentially we're getting convergence points being formed in a disticus phyllotaxy and the nice canalization of these veins as we move apically. So I'm going to run through how this model works in words, not equations, but behind all this is a whole series of differential equations. So first I'll tell you what up the gradient polarization actually means, and that's the idea that if you're a sister of pin 1, you are going to be allocated to the membrane adjacent to the neighboring cell with the highest auxin concentration. Okay? So already you should see that there's a positive feedback where if I'm this cell and I get a little bit of auxin, well then all the sister is going to be oriented towards me and I will accumulate more auxin. And there's a positive feedback that will create these convergent polarization patterns. So there's going to be one right over here, somewhere, boom, like that. Okay? Now pin 1A and pin 1B are allocated with the flux. And what that means is that if I have flux of auxin through this membrane, I will place more pin 1A on that membrane, and there's a positive feedback on flux. Very much like, again, like erosion. Pin 1A and pin 1B are simply allocated to different degrees with the flux. Okay, so I'll just say right here that there are no proven molecular mechanisms for sensing flux. There are no proven molecular mechanisms for sensing the concentration of auxin in the neighboring cell. But to me, as primarily an experimental biologist, what this did is say, aha, these two clades might be fundamentally different with respect to the machinery they're interacting with that polarizes them with respect to auxin. And one of the specific hypotheses from this was the presence of this cell. Okay? So here's my maxima, here's my lovely convergence of sister of pin 1, here's my vein moving down into the previous tissues right here, and here we have the presence of a cell where all the sister pin is oriented towards this membrane up the gradient, and all the pin 1A and pin 1B is oriented on this membrane with the flux. Okay? So the presence of a cell where we have bipolarity, the two different clades polarized in different tissues. And this took us a long time to work out experimentally, but suffice to say now we can place the two pins under different colors in my spikelet. Same thing's happening right here. And it's right around this maxima where morphogenesis is going to be happening, where that cell might exist, where these two clades are going to overlap. And I wouldn't be belaboring the point if it wasn't true. So here we're definitely pushing the resolution of confocal microscopy, but all I want you to do is look at this cell right here. Here is where the maxima is going to form. There's that periclinal division. And I don't think it's too hard to convince you that on this membrane, you have sister, and sister is still oriented towards that maxima, whereas on this membrane, you have pin 1B, and pin 1B is oriented away. So we really do see a cell where the two clades are polarized in different directions. And to me, this is a big clue, because this means that expression context doesn't really matter, the location in the plant doesn't really matter. There's some sort of protein intrinsic features between the two different clades that define their polarity in a single cell. And we've spent a lot of time trying to set up genetic assays to get at those protein intrinsic features. So one of the first ways, I mean, I really consider myself a geneticist, but for years we had no mutants and in Brachypodium, but CRISPR has been the miracle that everybody said it would be, and we were able to acquire mutants in all three different genes in Brachypodium. And the question here is, are their functions really different in Brachypodium? I'm not going to get too into the phenotypes quite yet, but suffice to say it was very cathartic that given our ideas about what sister was doing, creating convergence points, making the maxima, and that the maxima was necessary for organ initiation, sure enough, when you have sister mutants, you end up with barren pin-formed meristems. Here's a wild type. PIM1A, on the other hand, and PIM1B really do not affect organ initiation. So here you can see a single mutant of PIM1A, PIM1B, and a double mutant of PIM1A and PIM1B. There are phenotypes here, which I'll come back to, but suffice to say, organ initiation was not affected. And I think this is really cathartic, important validation for a lot of the ideas in Arabidopsis, that it's the convergence of pins in the epidermis that was really important. Okay, so another way that we've been testing the difference in function between these two different proteins is to use Arabidopsis as a test bed, a functional assay. Here is that PIN mutant, PIN1, and this 613 allele is a null. It makes no protein. The question is, when you express sister of PIN1 and PIN1B under the Arabidopsis PIN1 promoter, which one will be able to mediate organ initiation? And it's perhaps not too much of a surprise that sister can mediate organ initiation, whereas PIN1B can't. These plants are phenotypically indistinguishable from the null allele alone. Sister, I need to point out here, and this will become important much later in my talk, while it can make organs, it looks like more or less a normal plant. It seems to have trouble making anthers, and it's sterile. Okay, so I don't recover seeds from any of these plants. But suffice to say, again, there's some sort of, these are both under the same transcriptional control, there's some sort of protein intrinsic features that are different between the two. What are those features? Well, for that we need to look at the localization, but first I need to show you what Arabidopsis PIN1 looks like in some of the tissues we're going to be looking at. So here is, here's an SEM of Arabidopsis that I've shown you before. When we image this meristem, we look down on top of it, and it spins out flowers and organs in a spiral phyllotaxy. So that's this here, and you can see right here this convergent polarization of Arabidopsis PIN1 of those organs being formed. It's very abundant in the epidermis, but also very abundant in the vasculature below the surface. This is a stem that's essentially been sectioned in half and then split open and put on the confocal. So one of the biggest clues for why PIN1B was unable to complement came from when we looked at sort of the tissue level abundance, and that is when you actually look for PIN1 in the meristem, there's very little protein abundance at all, and in fact when you split the whole plant in half, it seems that it doesn't accumulate in the epidermis at all, whereas sister accumulates in the epidermis quite fine. And I'll just remind you that these are under the same exact promoter. This is pointing to a tissue level stability that seems to be protein intrinsic. So then we tried to really just shove PIN1B into the epidermis as hard as we could, and for that we used a two-component system driven by this ATML1 promoter, which is really highly epidermal enriched. With this system, sister does the thing you would expect it to do, creates lovely convergent polarization patterns, nice epidermal accumulation, but with the exact same transcriptional control, PIN1B looks like blobs. Okay, it's kind of unclear what these blobs are at this point, but it's likely that PIN1B is being targeted to the vacuole. There's certainly precedent for pins being ubiquitinated and targeted to the vacuole. I think what we're seeing here is PIN1B actively being cleared from the epidermis when we are really transcriptionally upregulating it in that tissue. So obviously, this is going to provide a barrier to mediating organ initiation. And just to show you what PIN1B looks like during the complementation, here's this big, long, barren pin meristem and a series of sections and then some details of what PIN1B is doing. You can see that it doesn't accumulate in the epidermal tissues very well, but just below the surface where this vein is forming, you get nice basally polarized PIN1B, really good protein accumulation below the apex, but suffice to say, PIN1B is not sufficient to mediate organ initiation itself. Okay, so I'm going to summarize what I've shown you so far. First is that with respect to this small group of pin proteins, Arabidopsis is really unrepresentative of most angiosperms, which contain this sister clade, which is present in Brachypodium. When you look at the polarization dynamics of the sister versus the pin 1 clade, there is single cell differences in polarization, which really points to protein intrinsic features that are different between the two clades. They have different functions in Brachypodium. And then when we test in the heterologous system the differences between the sister clade and the pin 1 clade, there's these tissue level differences in tissue level accumulation that have functional consequences when we try to complement the null allele. So this, this talk is really a setup for a lot more experiments. Now we have these genetic assays. We have 2 proteins that we really feel like have functional differences. We have these polarization behaviors that are combined in Arabidopsis pin 1, but split between 2 different clades in Brachypodium. And we can really learn a lot about what controls polarization. Now, I'll grant I haven't really told you what controls polarization. We still don't know. Clearly, one thing that we need to hammer on is in order to polarize convergently, the protein needs to be stable in the tissue where convergence points happens, and that's something that we can get back to as far as like what is controlling tissue level stability of pin proteins. And that's really not something that is talked a lot about with pin proteins, I think, in the literature. Okay, so this is kind of the bread and butter for my lab's pin work, and I do want to show you some of the directions that we're going with this, and they can sort of loosely be divided into these 3 categories. One is now that we have a model system that actually has these 2 clades, what are the wild type functions? Okay, so there's a whole bunch of functions of Arabidopsis pin 1 that are genetically intractable because when you mutate pin 1, you make no organs. So now that we have the 2 clades, what is happening in Brachypodium, and I'll tell you a little bit about how pin 1a and pin 1b are actually controlling plant height through the internode. The other is still maintains this question of what controls pin polarity. Part of this, it will be to leverage more Brachypodium imaging and quantitative imaging, drug treatments, all kinds of other assays for looking at what is happening in these cells where one pin goes one way and the other pin goes the other way. The other is to take a look at the proteins themselves in our complementation and our functional assays that I've established here, and try to define what are the domains that might explain why one polarizes one direction versus the other. And I'll show you one experiment from this in a second. And the other is to go back to evolution. My background, I have a pretty long history in evo-devo. I'm fascinated with the role of pin proteins in the acquisition of complex multicellularity in the plants, and I think that there is probably a very important role for these proteins in that. Also, I'll show you one experiment to me that suggests that pins might be a target for morphological evolution, and I'm very interested in exploring that from an evolutionary standpoint. So these are sort of broad brushstroke pictures of some of the things that I'd like to work on, but I'll just show you one experiment from each category now just so you can get a flavor of where we're heading. So this has to do with what are the phenotypes of pin 1a and pin 1b. I already mentioned that they don't have organ initiation defects, but the biggest phenotype that we can see with these plants is an effect on internode growth. So these are all the distance between the various organ initiation events in a series of genotypes. You've got pin 1a, pin 1b, and then a double pin 1a, pin 1b mutant. Pin 1b ends up with very long internodes at the base. And you can see that here. So here's these are 2 analogous internodes. These are 2 analogous internodes. And that results in an overall plant height of pin 1b that's a lot taller. Now, plant height is one of these things. It sounds boring, but it's tremendously agriculturally important. A lot of the targets for domesticating wheat in particular, which Brachypodium is really closely related to, concern plant height. So this is something we're very interested in following up on. The double mutant, fascinatingly, has a synergistic phenotype where it's very, very short internodes. So these are very, very small plants, but they make organs just fine. They set seed. This gives us lots of interesting experiments we can do. And I think those complicated dynamics have a lot to do with the bizarre expression and polarization dynamics that we're seeing in the internodes. So this is a series of internodes going down with pin 1b at the node and pin 1a mapped on top. So we've got to look at what's happening with auxin dynamics in this tissue, what's happening in the various genotypes, and this is very much focused on what is the native function for grasses in particular. Okay, down to tissue level, so polarization and tissue level stability. We've started chopping these guys up, really pushing hard on the transgenics in Brachypodium and the imaging in the spikelet meristem. And this is a very brutal experiment where we take this pin protein, and pin proteins have this transmembrane domains, a large loop domain, which has a series of modified sites, and another series of transmembrane domains. And we've simply chopped off most of the protein, all of these transmembrane domains, and a large part of the loop. It's kind of a cheap and wild promoter reporter. But what I just want to point you to is that normally in the wild type plant, there's very little sister accumulation internally, but with most of this information chopped off, you get quite abundant sister of pin 1 internally. And this again points to the idea that even sister might be regulated at the level of tissue level abundance. Normally, all of this protein is cleared because of these sites. So we can get increasingly more granular with this and try to really figure out what are the domains that make sister and pin 1a and pin 1b different. And then this is probably my favorite experiment of my career, because it's total happenstance, but I think it's really a window into pin biology. And that is that we got interested in taking other alleles of pin 1 and performing our complementation assay. So take pin 1a, or take pin 1b, take sister pin 1, complement this allele. This is called pin 1,4, which I say hypermorph here like it's a done deal, but nobody really knew this made protein until we did some immunolocalizations, and it turns out it makes protein. It's not polar. It's probably stuck in the ER because of this perinuclear localization, but that definitely needs to look. We need to look back into that. But a bizarre thing happened, and this is kind of a wrench in the whole narrative I've thrown you today, and that's that when you complement this allele with sister of pin 1 and pin 1b, both complement perfectly, okay? And by perfectly, I mean these make seeds this time, whereas when sister was complementing the null allele, we didn't get any seeds. Now we have full complementation. That alone is fascinating, but what is even more interesting is that if you'll recall, pin 1b in the epidermis is in blobs, probably being cleared to the vacuole. In the presence of this interesting pin 1,4 allele, pin 1b jumps on the membrane, creates perfect convergent polarization patterns, and it's presumably this that's actually able to fully complement. Now, I've thought a lot about this, and I have no better explanation than direct pin-pin interaction between this weird hypomorphic allele and this protein that has no ability to mediate organ initiation in the null allele. Pin-pin interaction is not something that has been studied in the pin field, and I think could be a real clue for how these complicated dynamics of inversion are happening. And that's because if I can return to this image, which is one of my favorite images, it's right around this spot where pin-pin interaction might actually be occurring. This is where morphogenesis is happening. This is where the inversion of polarity is happening. This is where all that maximal auxin is being plumbed in with the rest of the vasculature. So we've already started a series of approaches to try to test whether these two pins interact. And I think that could be a real clue for how pins work. Because I guess I didn't really get into that at the beginning, but they're fairly large families, okay? There are 9 proteins in Arabidopsis, and they all tend to have different behaviors. If one pin can communicate either polarity, stability, any number of different interactions with kinases, ubiquitination enzymes, etc., then that is going to be very interesting in these tissues where pins actually overlap. Okay. So the last experiment I'll show you is kind of a pie in the sky idea, and that's that when we perform this complementation of that interesting pin1-4 allele with sister and pin1b, the plants, while I said the complementation is perfect, the plants are phenotypically pretty different. And the most clear difference is that when you complement with pin1b, the stems are really thick, okay? They're very thick stems. Despite that, the vasculature is perfectly formed, the phyllotaxy is perfectly normal, and yet we have a morphological difference. And this has led me to the idea that, hey, over evolutionary time, maybe changing the relative contributions of, epidermal convergent maxima formation versus canalization throughout the tissue, throughout evolutionary history, maybe that's a source of morphological evolution and diversity. So we're taking a whole bunch of approaches in Arabidopsis to try to manipulate the relative contribution of sister and pin1b to see if we can manipulate even subtle aspects of plant morphology to see if that can be true. Okay, so I've come to my acknowledgments, and this is a cool acknowledgement slide because it's essentially a history of my scientific life on this project. This started in the lab of Sarah Hake, and she really let me go very much off-piste because this, as Siobhan mentioned, we primarily used maize in Sarah's lab, and my brachypodium work came out of a side project that's just sort of expanded, and then Audeline basically said, I'll fund you to keep doing what you're doing, brought me to the lovely Sainsbury lab, and has funded my work ever since. And then from the Leiser lab, I was very lucky to be asked to apply and get one of these career development fellowships, which has really allowed me to continue this linear trajectory here at the Sainsbury lab. And Sam is my technician who's over there in England still pushing things through on the transgenic fund. So there's, of course, my collaborators that I need to thank, but really these two seminal labs have been a huge part of about facilitating this whole project moving forward. So with that, I'll thank you very much and take questions.